Okay, in this video we are going to talk about ethanol metabolism. And I'd like to break this into two distinct parts of ethanol metabolism. First, let's go over all of the reaction sequences to see exactly what kind of chemistry is going on with metabolizing ethanol. And then talk a little bit included in that about the usefulness of what the products are of ethanol. And then the second part of this, I'd like to discuss exactly what's happening following the ethanol metabolism in the physiological state and the alterations in energy balance that occur and lead to disease processes. Okay, so first step is our ethanol. So ethanol comes in a variety of forms. I think most people are familiar. Wine, beer, a little harder stuff, margaritas. So the question is ethanol bad? Well, that's debatable. In excess, of course, ethanol is bad. Ethanol, anything in excess is bad for the body, but we also read about individuals who increase their lifespan by consuming a small amount of ethanol each day. So there's some active research going on ethanol metabolism in the body. Now, the first step in ethanol metabolism is by the liver. So the liver is the main organ that is going to be processing the ethanol and breaking it down. So the liver takes up the ethanol, and the first step of the reaction is going to convert the ethanol into an aldehyde structure. So in doing this, we are going to generate from ethanol to the compound acetaldehyde. And in this process, we are going to generate, in the process of oxidizing ethanol into acetaldehyde, we are going to be generating NADH. So we're going to be generating energy. And the enzyme that does this is something I'm sure most of you have heard, is alcohol dehydrogenase. So this brings me to an important point that in the old days, a lot of people thought that ethanol itself contributed no caloric content to a diet, and therefore it was empty calories. Empty calories in the form that it didn't really contribute to energy consumption, so it wasn't really something you had to worry about in the diet. But on the first step of ethanol processing, metabolism, look at the ethanol here being converted to acetaldehyde. We are producing NADH. And the NADH, of course, you know, is an energy-carrying substrate. Now, once we form the acetaldehyde, the next reaction that we are going to process is to take the acetaldehyde and convert this to acetate. Now, once again, notice that this is a redox reaction. So we are going to convert more NAD into NADH. So the second step of ethanol metabolism is also producing additional levels of NADH. This enzyme is acetaldehyde dehydrogenase that performs this reaction. And we are taking ourselves down to acetate in this process. Now, the acetate is going to contribute more energy because the next step of this reaction is that acetate is actually, in this process, a reasonably inert, harmless molecule. So the acetate can be sent out into the bloodstream, and where muscle can take it up and use it as an energy source. Alternatively, the liver can keep the acetate and convert the acetate into a molecule that I think everybody is now very familiar with. And this is, of course, acetyl-CoA. The enzyme that performs this is acetyl CoA synthetase. Now, this is happening in the liver to generate acetyl-CoA, but the muscle can also do this because muscle also has this enzyme. So muscle can also convert acetate into acetyl-CoA. So ethanol can be used for an energy source for both liver and muscle. Of course, then you know what happens to the acetyl-CoA. This will, of course, depend upon whether you are in a fed or fasting state. So let's say that we are in a fed state, which means that we are consuming our alcohol with food, say that nice glass of wine with dinner, or that margarita with our fajita plate. And then the acetyl-CoA can be used to produce energy, but remember what happens in the energy production cycle in the liver when our energy charge in the liver is high, which means we have high levels of NADH, high levels of ATP, our glycogen stores are filled because of our carbohydrate consumption. What happens to the acetyl-CoA in a fed state in the liver? Well, remember the acetyl-CoA carboxylase to make malonyl-CoA, and then this is going to be used for fatty acid synthesis. So fatty acid synthesis is going to be the main endpoint of alcohol metabolism in a fed state when the liver reaches a high energy conduction level. So this is where ethanol will increase our fat load in our body. This is the process and how it happens. Now in muscle, of course, the acetyl-CoA is going to be used for energy production, so that'll feed directly into the TCA cycle. Now, in a fasting state, this is where we get into a little bit more of a problem. And what I'm going to talk about now is the problems with alcohol metabolism in a fasting state, because this is typically more medically relevant, and it leads to a lot of metabolic disorders and metabolic imbalances, and this is something that alcoholics tend to face. The key here is that we are going to generate a very, very high energy charge. So even though we're in a fasting state, we're consuming alcohol, which generates an enormous amount of energy. We have two NADHs that we're generating from alcohol metabolism, ethanol down to acetate, and then the acetate converted to acetyl-CoA, which goes into energy-producing pathways to generate even more energy. So when we're talking about alcoholism, we're talking about underfed, poorly fed, poor nutrition individuals that are consuming large amounts of alcohol, leading to this strange high energy state, which is going to lead to various forms of disease. So now let's take a look at that on our next slide. Okay, now the first step that we have to consider is the oxidation of ethanol in liver to generate acetaldehyde. This reaction is happening in the cytoplasm. What we're generating out of this reaction is NADH. One of the problems with NADH is that if we have a sufficiently high level of NADH, we have to transport the NADH into the mitochondria. There's two issues of how NADH is going to be handled under these conditions where there's a large amount of ethanol consumption. The first one you are aware of, and this is the transport of NADH reducing equivalents from DHAP to glycerol 3-phosphate. And in this process, remember this goes back and forth, this is a dehydrogenase process, And this process here will transfer the electron equivalents to the mitochondria. So this is the normal pathway for getting NADH from the cytosol into the mitochondria, into the electron transport chain for energy production. Now the alternative way is that if this pathway becomes saturated, then the NADH is going to take another path, because if we don't have NADH oxidation, we are going to be stopping any type of glycolytic activity we're going to have here. So, with high levels of NADH starting to build up in the cytoplasm, what we are going to have now is the conversion of pyruvate to lactate. Now remember, this is also a redox reaction, but we're going to oxidize NADH to NAD, generating large concentrations of lactate. Lactate now, from the liver, can go into the blood. This is a problem calling lactic acid or lactate acidemia, because what is happening in the blood? We are dropping the pH. So the pH will decrease under large alcohol consumption because we are generating lots of lactate in order to regenerate our NAD in the cytoplasm. This enzyme, of course, you know this one, is lactate dehydrogenase. So this is one of the first problems here in generating lactate acidemia, because we're dropping the pH in our blood. So in a fasting state, this is a huge problem for excess alcohol consumption. Now, the second problem that we need to concern ourselves with excessive alcohol consumption in a fasting state is the process of energy charge in the mitochondria. So, remember, in a fasting state in the liver, a fasting liver, we have beta oxidation occurring at a reasonably high rate, and this is generating a lot of acetyl-CoA. Now, problem with this is that we are also contributing to the acetyl-CoA pool with alcohol consumption from acetate. And the acetate will convert into acetyl-CoA by acetyl-CoA synthetase, increasing the pool of acetyl-CoA that's already high. Now remember, if we are considering ourselves in a fasting state with somebody with poor nutrition, such as an alcoholic, that acetyl-CoA concentration is going to build up so high in this fasting state that we are going to begin generating ketones. So remember, the first step of ketone synthesis is taking two acetyl-CoAs, combining them to form acetoacetyl-CoA by the enzyme thiolase. Do a couple more conversions and we've got our acetoacetate. And our beta-hydroxy butyrate, our two ketones, which now, because they're being constructed, they're being synthesized at a high rate, go into the blood. So they're providing an alternative energy source. Now, with constant ethanol consumption and constant ethanol metabolism, we continue to increase our acetyl-CoA concentration. We keep putting ketones into the blood, and what happens there? Once again, we decrease the pH in our blood. This process is called ketoacidosis. Acidosis. Now this one here has an outward presentation, remember, because the acetoacetate in the blood can spontaneously convert to acetone. So this is why sometimes you will, if you happen to be around an alcoholic that has uncontrolled ketogenesis occurring because they have not eaten properly and they've been consuming large amounts of alcohol, that they will begin to smell like acetone as they exhale, because the acetone is volatile and it's coming out of the lungs. This is a clear indication that if somebody smells like acetone, that they are most likely in what's called a ketoacidotic state. Now the other issue that we have to concern ourselves with is the issue of hypoglycemia with alcohol consumption in a fasting state. Now how does this happen? Well, recall that with ethanol oxidation, we are generating large amounts of NADH in the cytoplasm, and this NADH now is going to begin to convert pyruvate to lactate by lactate dehydrogenase. So this is the reaction that we saw just a few minutes ago with how the NADH levels are being handled in the cytoplasm. Now, the question is here is how is the pyruvate going to be converted to oxaloacetate to malate to the cytoplasm and contributing to gluconeogenesis when the pyruvate is being converted at a high rate to lactate. So precursors coming in to the gluconeogenic process, such as, for example, the amino acid alanine, because remember, we're in a fasting, probably more of a starvation state, because this is an alcoholic who isn't eating properly. This alanine comes in, the transamination reaction of ALT converts the alanine into pyruvate. Now, under normal circumstances, the pyruvate will be converted into metabolites, leading to gluconeogenesis. But with the high NADH level, our conversion of the pyruvate is going to be disrupted. It's going to be redefined into lactate. And then, of course, the lactate decreases our pH in the blood by increasing its concentration in the circulation. So this is a way that we can inhibit gluconeogenesis with high levels of ethanol production. And remember that in an advanced fasting state or starvation state, this is the primary source of glucose. Primary generation metabolism of formed glucose, because at this point our glycogen stores are depleted. Now, let's shift back to the fasting state, the fed state in the liver, just for one more piece of information. The fed state in the liver, if we're taking our ethanol and we are oxidizing this ethanol, remember we are going to be forming in the liver acetyl-CoA, will be our end product. Well, if we have high concentrations of acetyl-CoA in a fed state, remember that this is largely going to be forming fatty acids. So we'll be performing fatty acid synthesis. Now, as I said, the ethanol will increase our fat consumption, but our fat production, but the fatty acids then have to be converted into triglycerides in order to be stored in adipose tissue. So in order to do this, we need to add the backbone structure of triglycerides, which is, recall, glycerol 3-phosphate. Now, where does this glycerol 3-phosphate come from in order to continue the process of triglyceride synthesis? Because we're generating plenty of fatty acids. Well, remember these NADHs in the cytoplasm. From that first step of ethanol oxidation into acetaldehyde, the acetaldehyde, of course, will enter the mitochondria at that point. But this first step of NADH, remember that we are going to be converting DHAP into glycerol 3-phosphate by the enzyme glycerol 3-phosphate dehydrogenase. So this is a method that we talked about a few minutes ago in a way to translate the NADH from the cytosol into the electron transport chain in the mitochondria. But we are generating a large concentration of glycerol 3-phosphate. From this reaction, we have abundance of NADH in the cytoplasm because of the ethanol we've consumed. And so this glycerol 3-phosphate can go in two directions. It can either be used as the backbone structure to construct triglycerides by adding our 3 fatty acids on there, and then it gets transported to adipose to increase our fat load, our fat storage, or it can be converted back into DHAP in order to transfer those electrons from the cytoplasm into the mitochondrial energy production. Now, the energy production in the mitochondria, you can imagine that we've got so much NADH flooding this electron transport chain that we're probably at such high energy state that the electron transport chain is going to be inhibited. So this direction into glycerol 3-phosphate is going to predominate, leading to increased triglyceride synthesis. And here's how we can consume excessive amounts of ethanol in a fed state and how it can add to our fat storage capacity. Okay, those are the main features that I want to cover of ethanol metabolism, and this is pretty much the basis of what will be on the exam. So in class on Monday, I'll cover a little bit more of the generic regulation steps on here, and a few more other little items of how ethanol can increase our toxicity in the liver and overcome some of our drug metabolism abilities.